Alright, so in this video I'm gonna try to explain to you how DNA replication works, and I'm gonna try to break it down step by step so that you're gonna understand like how we get leading strands, how we get lagging strands, what are Okazaki fragments. And by the way, I'm gonna mention some proteins and enzyme, but I'm not gonna go into detail about it. I'm gonna make a separate video so that it's easier to understand this concept better. Alright, so in the lecture last Thursday, or in the first week, we had something like this. We had an activity where there's an origin and we had to draw the new daughter strands according to the instructions. I just want to start first by showing you the final answer. So our final answer should look like this because we have a leading strand over here. It's 5' to 3', and a lagging strand on this side. Same way on the bottom, we have 5' and 3' on this side. It's a lagging strand, and then this one is a leading strand. So that's what I'm gonna try to show you, is like how do we get the leading strand and how do we get the lagging strand. Alright, so let me start with showing you how to do it. Alright, so as mentioned in class, we know that DNA is double-stranded. And double-stranded DNA is anti-parallel to each other. So that means if DNA is double-stranded and it's anti-parallel, I can draw something like this. Right? So if it's let's say one of the top strand is 5' to 3'. And they're anti-parallel to each other, which means the bottom should be 3' to 5'. Alright, so the direction of the strand is always starting at the 5' to 3'. So the direction of this should be going to the right, while the direction of the bottom strand is going to the left because it's 5' to 3'. Another important thing we need to know is that nucleotides only add at the 3' end of a growing strand. Let me write somewhere around here. This is one of the important things that we need to know, especially in biology. It doesn't add on the 5' end, it doesn't add on the 2', 1', 4'. It only adds at the 3' end. It has something to do with DNA polymerase and its proofreading ability, but I will not be going into detail about that. You'll be learning that in cell biology or higher level classes. Alright, so let's start on the activity. So, in the activity, we had something like this, right? We have an origin over here. So let's say, so we know that origin is gonna have a replication bubble. It's gonna cause a replication bubble to open. So that means it's gonna look something like this, right? You have something like this, and then something like that. It's gonna be small at the very first few minutes. Let's say it only started. This one is only started, and I'm gonna try to zoom this in. I'm gonna make this bigger, just to show you like how the daughter strand is gonna look like. We have a imaginary line for the origin. Alright, so one trick that I use to make it easier for me is that I can imagine that this imaginary line of the origin is like the end of a strand. So for now, I'm gonna focus on the top strand. Everything is gonna be about the top strand for now. So let's say I wanna make this easy for me. I know that this is the end, so that means if the beginning is 5', the other end should be 3'. So I could say that this is the 3' end. And then on the other side, I know that this is a 3 prime. So that means the beginning end of the strand should be a 5 prime, right? This will make it easier for you. You don't really have to do this, but I think it works really well. Alright, now that I kind of labeled this, I can start making my daughter strands. So, as we know, strands start from 5 prime to 3 prime. So that means if I'm gonna have a new daughter strand, I'm gonna have to start from 5 prime. And we know, let's say, and we're only gonna use the top strand. So the top strand is gonna be our template. And since strands are anti-parallel to each other, that means the daughter strand has to start over here at the 5 prime. And then it ends at a 3 prime end. You see how the top strand is like anti-parallel to the daughter strand? Same way over here. I know that this is the 3 prime end, so that means I have to start at the 5 prime end over here because it's gonna be anti-parallel. And it's gonna end to a 3 prime. Now, you might think, why is it so different to our final answer over here? See how this is a lagging strand? And the strands over here are just like one small strand, one strand each. It's not fragmented. So this is where the concept starts. So imagine that this is only like the beginning of the replication. And let's say a few more minutes pass by. And let's say the replication bubble got bigger again due to helicase unzipping more of the DNA. So it's gonna get bigger. Oh. We have the imaginary line over here. So since it got bigger, that means it only got bigger, right? It didn't get rid of the old strands before. So I can just copy this over here. We already have this strand over here. And then another strand over here. Alright, so as I said earlier, nucleotides only add at the 3 prime end of a growing strand. So that means in this daughter strand, I have a free 3 prime end over here. So that means I can keep adding. So I'm gonna just keep adding and adding and adding and adding until I get to this unopened replication fork. So now you see, this is one long strand. It's not fragmented because I can keep adding on the 3 prime end. But compared to this other strand, I cannot add anymore on this 3 prime over here because there's already a strand that's over here. So I cannot use the top strand as a template to make a new strand. So that means the primase has to add another primer somewhere else to continue replicating. So if I have a 3 prime here, now I cannot continue anymore. Now I have to start over here again. This is 5 prime, and then I'm gonna add to the 3 prime end again, and like that. So now you might see like there's like the pattern that I'm trying to show you. Let's say a few more minutes pass by again. Now the replication got way bigger again. I'm just gonna make this bigger. We have the replication origin here. Alright, so it just got bigger, so that means I can copy over what we had already. So we already have a 5 prime over here. And so let's say it got bigger, so it's gonna end around here. This one, we already have this one. Alright, so same way before. This one is a free 3 prime. So that means I can keep adding again on this one. Keep adding until I reach the end. Now I have a 3 prime over here. It's one long strand, right? Same way over here. I'm gonna try to keep adding here, but see how it's already like there's already another strand from the first few minutes. So that means I cannot add anymore on its 3' end. Now I have to add another strand starting here. So I hope it kind of like made you understand like why we had leading strands and why we had lagging strands. So if you compare it to our final answer over here, you see how we have lagging strands, same way as the way we did it. We have lagging strand on this way, and leading strand on this way, leading strand on this way, and lagging strand on this way. Alright, so I didn't do the bottom part because I kind of wanted you to do it. So pause the video right now while I'm doing it. Alright, so I'm gonna do the bottom strand now. Alright, I know that this is 3', so that means I can imagine that this is 5'. This end over is 5'. And I know that this is 5', that means it's gonna end at the 3' end over here. It's gonna make it easier for me to visualize what I'm doing. So I know my new daughter strand has to start from 5' to 3' again. 5', 3'. This other end, this is the 5', that means this is 3' over here. So I can start here. 3'. Right? Now I'm just gonna copy it over here. We already have something like that. Alright, so I know that I can keep adding on this 3' end. So that means I just can keep adding. And this is gonna be the leading strand. And this one I cannot add anymore since there's already a strand over here. So that means I'm just gonna have to start over here again. Same way again here. I'm just gonna copy what I have on the other side already. So I know that I already have a 5' to 3' over here. So I'm just gonna keep adding again, keep adding and adding and adding and adding until I get to the end, 3'. And then same way over here. I know that I already have 2 lagging strands from before. So I know that I cannot add anymore on this way. So now I'm gonna have to start on another. So yeah. So now, if you compare it to our final answer, it is the same. So this is like how DNA replication works. There's it's important to know that DNA replication doesn't happen on its own. There's enzymes and proteins involved in it, and I'm gonna make a separate video about it. So I hope that this works. I hope that it helps you understand why we have leading strands and lagging strands. In your exam, you're not gonna need to show all of this work over here. I just wanted to show you like how we get to the final answer, because in your exam you're gonna have something like this more so. I'm not doing the exam, but this is something like what you're gonna see, or what they would want you to put in your exam. And also go to the ALA and SI meetings. They have some ways to do this too. This is my way of understanding how DNA replication works, but Kenzie and Edita, they have some fun ways of describing DNA replication. So maybe it's gonna work out for you if you didn't understand how this works. So, alright, that's it for this video. Thank you.